Good evening, all. Am I audible? Hello. Am I audible?
Yes, you are.
Okay, so we are going to start the session. Wait for 2-3 minutes, then we will start the session. We are waiting for 2-3 more students. Okay. Okay, let's start the session. So this is week 1 microsensors implantable device and rodent surgeries for biomedical application. And this is a problem solving session. I'm Arpita Biswas, a PhD scholar from IIT Jodhpur, an electrical engineering department. And this is more details about me. So in this session, we are going to discuss about some of the MCQ question, and here I have taken some question, and we can discuss here. And also, if you have any other doubts, you can also ask here, or you can directly mail me. So in the next class, we can discuss this. Okay. So this is the first question. I think all of you have gone through the lecture video of this Hardik J. Pandya sir and the other faculty also is here, I think. So at first you have to go through all the lecture and then we can discuss the this type of question here. So this is the question number 1. Which among the following is correct for the sensor and systems? So first option is systems can be integrated to make one sensor. And the second option is the sensor can be integrated to make one system. The third option is sensors and systems are two completely different entities. And the fourth option is all sensors require systems. So go through the question carefully and try to give the answer. And at first we can discuss here what do you know about the sensor? What is sensor? Do you have any clear idea about the sensor? And what about the system? And there is no hurry. You take your time, then unmute yourself and try to give the answer. Anirudh. And I think one more student is here. Harini. Go through the question carefully and try to give the right answer. What do you understand about the sensor?
When system can be integrated to make one sensor. I think this is the right answer.
Uh, uh, can you please elaborately discuss about the sensor? What is sensor? What do you understand by the word sensor?
And sensor is a part of a system, but a system can't be a part of a sensor.
No, like what is the work function or what is the work, what is the need of the sensor, what it does?
Ma'am, basically sensor will sense any kind of change that is happening. Like
Mhm.
it can either sense a change in maybe light or maybe sound. So that is like any device that is sensing a change in the environment is a sensor.
Okay, actually, since we can, yes, it is little bit correct, right? Just a minute. Yeah, like any physical signal, like light or anything, touch, anything can be. Okay, any physical signals which a sensor it transfer this physical signal into the electrical signal, right? So, if you heard about the touch sensor or light sensor, what it does, you are giving some input in the term of like physical property, like light or touch, and then it convert this physical property into the electrical signal, and then we measure that electrical signal in the how it changes, like it's sensing. So, like in biosensor term, we are giving input like the like then biomolecules and then it changes its property or something. So there is an electrical signal changes. So this is called the sensor. Okay. Now coming to the question. Now coming to the point. The what is system? Any idea about systems? So you can easily can give the right answer. Like the sensor is a part of the system or system is a part of the sensor.
And sensor is a part of a system.
Yes, systems mean what do you understand? Like how you think when the word coming, it is a system. Like it is a whole things. Like in the system there is a small part of the part or sensors, and then it's making a proper system, right? It's the whole things. So what is the right answer here?
Ma'am, option two.
Yes, option B is the right answer. Sensor can be integrated to make one system. Multiple integrated means sensor and the other things or multiple number of sensor. Sensors can be integrated to make one system. So the right option is option B. Okay. Good. And the question number two is here. Which of the following is correct for experimental neurophysiology? So first option is the biopotential provide useful information about the health status of the brain. The second option is the microneedles are used for the electrode acquisition. Third is EEG is used as a primary metric for neural disorders, and the fourth option is all of the above. So go through the question carefully, take your time, and then try to give the right answer. And there is no hurry. Take your time, but at least try. Nikita, what is the right answer?
I'm not sure. Is it all of the above?
Okay. Another students, what is the right answer, Harini? Anirudh, what is the right answer? E-
Ma'am, EEG used as a primary metric for neural disorder.
Okay. So for your side, the right option is option C, right? And what about the microneedles are used in the depth electrode acquisition? And what about the biopotential provide the useful information about the health status of the brain? See, I'm not expecting that all of you are going to give the right answer, but at least try. Go through the option one by one carefully. Biopotential provide the useful information about the health status of the brain. Yes, this option is also true. Microneedles are used for depth electrode acquisition. This is also true. And EEG is used as a primary metric for neural disorder. So all of these are the right option. Okay, so the right option is here, option D, all of the above. Okay. So next move toward the question number three. So what is the correct flow for neural engineering research? So first option is cat, then human, then monkey, then rodent. And the second option is first rodent, then cat, monkey, then human. Third, rat, monkey, rodent, human. And the fourth is the rat, human, monkey, or rodent. So you have to find out the correct flow. And this topic is already covered in the lecture, I think. The first two lecture, the introduction 1 and 2, this topic is already covered. Yeah, Harini, option 2. Okay. Any other? Anirudh and Nikita. Okay, Devstuti. Option B. Harini, what is the right answer? Anirudh, what is the right answer? No, option C is not the right answer. Option B is the right answer. Okay, first rodent, then cat, then monkey, then human. So the correct flow is option B. Okay, so next move question number 4. Why are rodent preferred over the animal for advanced neural science experiments? So first option is due to the anatomical similarities with humans. Second, due to the genetic similarity with humans. Third, due to the physiological similarities with humans. And fourth is all of the above. So go through the question carefully. Why are rodent preferred over the animal for advanced neural science experiments? You can unmute yourself and give the right answer. Nikita, option A. Okay.
Ma'am, option D.
Okay. Can you a little bit explain?
Yes, ma'am. Like
Yeah.
anatomically, most of the organs and organ systems are quite similar to us if we
Hmm.
compare it with the rodents. Same goes for the genetic similarities because there is somewhat very little percentage of genetical dissimilarity. Other huge portions of the genetical parts are quite similar, and
Okay.
physiological similarities in cases like the systems that we and the rodents or any other animals that we share are quite same, like the reproductive system, the respiratory system, and all the systems. So I felt all the above should be the right answer.
Yes. Very good. The right answer is all of the above. Okay, and the explanation is also good. So next move toward the question number 5. Consider the following two statements about the ECOG and select the correct option. So first option is for first statement is ECOG and ECG are two completely different modalities. And the second statement is ECOG has better spatial resolution than scalp recorded EEG. So here you have to find out the right option. Like first option is the both is the statement one and two are correct. The second option is the both statement one and statement two are incorrect. And third is only statement one is correct, and fourth is only statement two is correct. So go through the question carefully and find out the right answer. Consider the following two statement about the corticography and select the correct option. What is the right answer? I think in the lecture also they have taught about the ECOG, right? And also in medical term you have heard about the ECG, where it is used. And ECOG is used, I think, for brain functioning, right? And where the ECG is used, so it is completely different in modalities or not. So is this statement true or false?
True, mom.
Okay, and what about the statement two? Like ECOG has better spatial resolution than scalp recorded EEG. Anirudh, what about the statement one? What about the statement two? Statement one is right, okay. And what about the statement two? Sudipti, Guru Raj. What is the right answer? At least try. Okay, so the ECOG has, okay, both a statement true, ma'am, because it's completely different. You can unmute yourself and discuss here, no? So it is a little bit helpful for other student also. You can unmute yourself.
Mom, what is meant by spatial resolution? Because I have a little bit of idea about how the both are different in their working principle. What
Hmm.
is meant by spatial resolution?
Like spatial, have you heard about the resolution? Like in fabrication term, what we understand about the resolution? Like in the fabrication term, when we are going for the sensor fabrication, resolution means like we are taking 2 points. Okay, now not in the medical term, in the fabrication term, I'm simply understanding like we are taking in between this 2 point. Okay, so how we can when we are going to like fabricate the device or something here. So in optical term, we are seeing now. So how it's completely visualized, like in accurately in resolution, how much it's like the resolution is. We can see the different point like in the term of like in the photolithography when also we are going for the like the how can we understand, see just a minute, is there any difficulty if I am going to understand it in the term of the fabrication?
No, ma'am.
Okay, just a minute. I think here's the point. Okay, is the point. Like in the photography photolithography, like when you want to like the form the electrode like this type. Simple, we are taking the source drain. Okay, we are going. We next we also discuss about the PVD, CVD technique and also the photolithography etching. This type of technique. This is used for the sensor. Okay, in the so used for to fabricate the sensor. So if we are going to the concept of the fabrication, how the resolution like here. Um, so how we can visualize like this point, like this 2 point. If your photolithography we are going, so how you can clearly see these 2 point that the resolution how much in the yeah, also when you are taking the picture, I think in that case also you know about the resolution, how the if the resolution is good, so your picture quality also good, right? You can directly see the point whatever we need. So here they are discussing about the spatial resolution. So now you can give the explanation about how this ECOG is better than the scalp recorded EEG. In the next class also I will discuss more about this spatial resolution. Okay. Now whatever I'm trying to discuss, it is in the term of the fabrication. Hello.
Yes, ma'am.
So now you can explain about this answer.
Ma'am, that means whatever like electrical signal is recorded,
Mm-hmm.
uh, it is better resoluted or better distinguished in
Mm-hmm.
ECOG than EEG.
Okay. And what about the first one?
Yeah,
First
that is
statement.
a correct statement.
Okay. So the right option is option B, right? The statement one and statement two, both are, sorry, both are correct. This option. This option is right. So next move toward the question number 6. Which PVD technique are used to make the neural implant? So have you heard about the deposition technique? Like in the sensor fabrication, we used some the deposition technique. And there is different type of deposition technique is present, like the PVD, the physical vapor deposition, and the next one is the CVD technique, like the chemical vapor deposition. So if you're interested about to learn about this deposition technique or deeply, so I will taught here. So now going to the question like which PVD technique, the physical vapor deposition technique, are used to make the neural implant? First option is the thermal evaporation. Second option is the E-beam evaporation, and third option is the sputtering, and the fourth option is all of the above. Now here we can discuss about different technique. Have you heard about this PVD technique before?
Yes, ma'am, in class.
Okay, so can you little bit explain here what is PVD and what about this thermal E-beam or sputting? What is the main difference between these PVD techniques?
Ma'am, the basic difference lies in the working principle. Like in thermal evaporation, directly heat is provided to the source metal, by which it evaporates and gets deposited into the substrate. But in case of electrical beam,
Hmm.
a filament is used, which is heated at a very high temperature, that creates an electron discharge, which is
Hmm.
uh, whose path is deflected by deflecting magnets. So which that is like how it is depositing onto the substrate.
Hmm.
So definitely the thermal evaporation limitations have gave rise to e-beam evaporation methods.
And what about the sputtering?
Ma'am, I have no idea.
Anyone, what about the sputtering system? Anirudh, anyone know about the sputtering system? Gururaj, see, as you told, that's the thermal evaporation. What happened here? Like here is the sample in the thermal evaporation system. Here is the sample, and it is there is also a rotation. Okay, because we want the uniform deposition, and here is the substrate. Sorry, the uh, whatever we want to deposit, the material is taken here, and here is the sample, like this material holder is here, and we are passing a high current through it, so it heated up, and this material is evaporate and then directly deposited on this material. It is in the thermal evaporation system. And what about in the e-beam evaporation system? There is a material, and then like electron beam is used to heated up this material, and then it's directly deposited on the sample. Okay, here is the sample. And what about the sputtering system? So there is a cathode and anode is there. Like if it is a sample, like the target, like if you want to deposit the gold, this is the gold target is here, and here is a sample like silicon dioxide sample, where we want to deposit this gold. Okay, so what we done here in the closed chamber, in the sputtering chamber, we directly flow the argon here. So in the presence of plasma, so it's bombarded this target, and some of the particle, like in nanoparticle form, it is deposited on the like sample. Okay, so this is the basic principal difference. It is heated up, then it evaporate and it directly deposited on the sample, and here we use the electron beam to heat it up, and then it is evaporated and deposited in the sample, and here we use the argon gas. Okay, so argon bombarded with the on this AU or something, the target whatever you want to deposit, and this some of the particle of the this material or material of the target is deposited on the sample. So this is three main difference, and these three all happen in the closed chamber. Okay, this is the thermal evaporation system, this is the e-beam evaporation system, and this is the sputtering. Okay, so now guess the right option. Which PVD technique are used to make the neural implants?
All of the above, ma'am.
Yes, the right option is all of the above. Because here you can see that the actually work is the aim is same, right? The grow the material something to using this PVD technique, and this thermal EV manufacturing, all these are the part of the PVD system, so we can grow the material or deposit the material using these three. So for the neural implant, we can use the thermal, E-beam, and sputtering, this all. Okay, so the right option is all of the above. So this is the question number 7. Select the simplified correct sequence for neural implant process. First option is the PVD, then lithography, then etching, then characterization. Second option is first lithography, then PVD, then etching, then characterization. And the third option is the characterization, PVD, lithography, and etching. And the fourth option is PVD, lithography, characterization, or etching. So go through the question carefully and then try to find out the right answer. Have you heard about the lithography technique? What we done here?
Ma'am, I just know a very little bit of this lithography. As in class, it was told it is used for patterning.
Okay. And anything else?
No, ma'am.
And what about the etching process? The word etching. So, what do you understand from this word also? What is etching?
Ma'am, removal.
Yes, remove something we don't want or unwanted something. And what about the characterization technique? Have you heard about the characterization?
No, ma'am.
Okay. So characterization, like there is a two type of characterization, like electrical characterization or material characterization. When we deposit some active layers, so first we have done the material characterization, like there are different type of material characterization. I think it is apart from this topic, but if you are interested, I'm saying that like the XPS technique is there, like the EDX mapping, also the FESM image. So how the material, the active layer is growing. So in that case, we are doing the material characterization. Okay, the Raman spectroscopy, this type of material characterization. And when we go for the like device fabrication process, in that case, after fabricating the device, we check na, whether the device is working or not. In that case, we are going through the electrical characterization, like we check the device current or something, like we are giving the input, and as we are getting the changes or device is working fine or not. Okay, so this type of characterization, this is called the electrical characterization. So now, and what about the etching process? Etching is explained later, but first we are coming to the lithography. So litho is light, and graphy something stone or something. So lithography means the pattern transferring process, right? See here. Yes. Like if there is a this type of electrode is here, as I have shown earlier. This is the source and this is the drain. Source and drain, right? This is the electrode pad something. So if you want to like have you seen the sensor? Like here a source and drain is present and this is made of the gold. Okay, on the silicon dioxide wafer, and then we have make the source and drain. So in that case, there we use the mask. Okay, in the mask there is some pattern is here, like this type of pattern is in the mask. Okay, then using this mask, we pattern this like we transfer this pattern onto the substrate like the silicon dioxide. So this pattern transferring process is called the lithography process because there is a mask and there is some structure is there. So pattern is there. So we transfer this pattern onto the sample where we want this electrode. Like there is a silicon dioxide wafer or hemp wafer. So we directly transfer this pattern onto that. Like we pattern the source and drain gate something. Okay. So this process is called the lithography. And next, later we can discuss more details about this lithography, how we did this lithography technique. There are two, three, like if we are going for the yeah, optical lithography process, there are two, three types of optical lithography also, the contact printing and projection something. So next we can discuss in more details. Okay. So now just there is some small idea about lithography that this is the pattern transferring. So in the marks, there is some pattern is present. So we are directly transferring this pattern onto the sample or onto the sensor where we need the source gate and the drain. Okay. And now coming to the question, the etching process. So PVD is a deposition like the whole in the whole sample I have deposited the gold. Then I have transferred this pattern from the mask to here. I have pattern that I have transferred the pattern. So this much portion I want, and but I grow the sample in the whole. Okay. So I don't want the extra like gold here. So what we will do? We will etch now. This unwanted portion we will etch out. So this is called the etching process. And also next in the next time we can see how this the wanted person like the wanted portion of this electrode will stay and unwanted portion of the gold will remove. So this is called the etching. Okay. And then we go for the characterization like this device is working fine, this sensor is working fine or not. So now you can give the right answer. Which option is the right?
Ma'am, option A.
Yes. First, we have to deposit. So it is like deposit like the thermal evaporation or sputtering or any like EVM evaporation system. Then we have to go for the lithography process. So we have to transfer the pattern. Then the unwanted portion will we have to remove. So this is called the etching process. And after the etching process, when the device is done or the sensor is done, then we go for the electrical characterization. So the characterization also, there are two types like material characterization or electrical. Material characterization, when we in the sensor also we need some active layer in the actually different type of sensor, like if we are only taking some like chemi resistive or something, this type of device, in that time we'll have to grow the material. So this material also we have to go for the material characterization. But here if we are preparing the device of sensor, we have to go for the electrical characterization. See the it is if there is a pressure sensor, so you are giving the pressure or force on the particular area, and you have checked the if there is a changes of this device or the sensor property, right? This type of. So the right option is option A, the PVD lithography, etching, and then characterization process. So next move toward the question number 8. Find out the components of DBS system. First is the electrode, the second is the lead, and third is the stimulator, and fourth option is all of the above. So which one is the right answer? And at first tell me what is the full form of the DBS system. Take your time, but at least try. So what is the right answer? See, the DBS full form is deep brain stimulating. Okay. So find out the components. Now they are asking for the component of this DBS system. Electrode, lead, and neuro stimulator. So the right option is all of the above. In the lecture also they have seen the component of the DBS system. So this is the question number 9. Which is the common location in the brain chosen for the deep brain stimulator? So first option is the motor cortex, second is the somatosensory cortex, third is the subthalamic nucleus, and fourth is the occipital lobe. So which one is the right answer? There is no hurry, take your time, but at least try. Unmute yourself and try to give the answer. Here we are discussing this type of MCQ question because I think it will help to solve your assignment also, and also in the exam this type of question also come. So I think in the per week we are discussing about 30 to 35 MCQ question. It will cover almost the lectures, and I think it will be helpful for all of you. And if you want to discuss something, a particular portion, you can mail me directly, okay? So in the next class also, we can discuss for 10 to 15 minutes about this topic, whatever you want to discuss. So the right option is option C, subthalamic nucleus, okay? So next question is question number 10. Which part of the brain is visual perception in the human related to? First is the frontal lobe, second is the parietal lobe, third is the occipital lobe, and fourth option is the temporal lobe. So go through the question carefully, find out the right answer.
Now it's occipital lobe.
It is option C, right? Occipital lobe.
Yes, ma'am.
Okay, okay. And what about other students? Anirudh, what is the right answer? And I think Nikita is also here. Nikita? Gururaj.
Um, Ma'am, it is occipital lobe.
Okay. Gururaj, what is the right answer? Please unmute yourself and try to give the right answer. Yes, it is right. It is occipital lobe. Good. And this is the question number 11. What is the primary purpose of microsensor in biomedical application? So first option is this is the some easy question I am discussing here to just recall all the three lecture. Okay. I think all of you can give the answer. So, the question is, what is the primary purpose of the microsensor in biomedical application? First, to enhance the imaging resolution. Second, to monitor the physiological parameter. Third, to perform the surgical procedures. And fourth, to reduce the size of the medical devices. So, go through the question carefully. This is a very easy question. I think all of you can give the right answer. Please unmute yourself.
Is it to monitor physiological parameters?
Yes, yes. When we are discussing about the sensor, now at first I have told that what is the work of the sensor? Like the physical signal, it's converting the physical signals into into the electrical signal. This is the purpose of the sensor. So microsensor, the dimension is in the micrometer range. Okay, so in biomedical application also, its main work is to monitor the biological, the physiological parameter. Okay, good. So right option is option B, to monitor the physiological parameter. So this is the question number 12. What advantage do implantable device offer over the traditional medical equipment? First option is the reduced cost. Second, the real-time monitoring and localized treatment. Option C is the increased durability. And option D is the improved non-invasive diagnostics. So what is the right answer?
Mom, option B, real-time monitoring.
Yes, good. The real-time monitoring and localized treatment. So this is the question number 13. Why are rodent surgeries commonly performed in biological research? First is the rodents are easier to handle than other animals. Second, rodent have biological similarities to human. Third is the rodent require the minimal care. And fourth is the rodent are cost effective to use in labs. So what is the right answer here?
Answer B. Rodents
Okay.
have biological similarities to humans.
Yes, option B is the right answer. Because rodents have biological similarities to humans. This is a question number 14. Which of the following is a key challenge in developing the implantable devices? First is the ensuring the compatibility with imaging systems. Second, the achieving the biocompatibility and minimizing the immune response. Third, the reducing the size of the sensor, and D is increasing the battery life. So, what is the right option here?
Mom, is it achieving biocompatibility?
Yes, the first they are seeing that the biocompatibility. Okay, so the right option is option B, achieving the biocompatibility and minimize the immune response. So this is the question number 15. What is a common application of implantable microsensor in rodent? So first option is the behavioral studies. Second, monitoring the brain activity or metabolic changes. Third, the drug delivery, and fourth is the enhancing muscular strength. So find out the right answer.
Monitoring brain activity or metabolic
Yes,
changes.
yes. Actually, in the lecture three also you have seen that they are all discussing about the brain. Okay, the brain signal, brain monitoring, all these things. And here also we are discussing about how the brain signals of the rodent like their. So the right option is the option B, the monitoring the brain activity or the metabolic changes. Okay. So next question number 16. What technology is often integrated into the microsensor for biomedical application? Firstly, the wireless communication system. Second, the advanced surgical tools. Third is the CIRSPR gene editing techniques. And option D is the optical imaging system. There's different tools or different techniques they have used. So, what technology is often integrated into the microsensor for biomedical application? See, in the recent, like in recent years, what all of you are using the wireless, right? So in the microsensor also they are talking about this wireless communication system. Okay, so the right option is option A, the wireless communication system. The technology is often integrated into the microsensor for biomedical application. It is the wireless communication system. So
Mom,
next, Yes.
Excuse me, ma'am.
Yes.
In case of microsensors, like how can we connect it with wireless communication? Like that will just sense the signals or the waves, and that will give the output. And like how are we connecting it with the wireless communication system?
Actually, in biomedical or application, normally, like I'm in working also the fabrication lab. So what happened in that case, like in when we are going for the sensor fabrication, first we fabricate the sensor, then we tested it for like different like for gas sensor we are giving the input as a gas, and for biological like ascorbic acid, vitamin like vitamin C, that type of, when we are dropping in the sensor, so we are getting the output in the electrical term, electrical signal. Okay, and now we can convert, like some of them are using the like the Bluetooth system, like or the IoT system. So they are integrating this type of things with the sensor. So this is, that's why the question is, what technology is often integrated into the microsensor for biomedical application. Okay, so wireless communication system, so it will be good. So we can just we inject a micro needle in the brain or something in the body, and we can if it is a wireless communication, so we can easily see how it is working, and there is no connection is also required. So they are thinking about this type of technology. Okay.
Yes, ma'am.
So this is question number 17. What is the significance of minimally invasive rodent surgeries in biomedical research? So you have heard about this word, the minimally invasive, or in the lecture, I think two, right? If you've gone through the lecture, I think they have discussed about what is minimally invasive. Can you give an example about the minimally invasive or invasive technique?
Ma'am, invasive is something where we like implant something inside the brain.
Mhm.
Minimally invasive is where like a minimum invasion is done, and
Yes.
non-invasive techniques are where like we do not have to implant something or input something into the body.
Can you give an example of the minimally invasive technique?
Ma'am, is it TMS?
Have you heard about the glucose sensor, like just pinch, then some blood are coming, and then
Glucometer.
tested it, right?
Yeah, yeah, the glucometers.
Yes, yes, the glucometer. It is called the minimally invasive, not implant in the body. Some touch and some pin is here, and blood is coming, so it tested. Okay, this is called the minimally invasive technique. So now coming to the question, what is the significance of the minimally invasive rodent surgeries in biomedical research? First option, to ensure the ethical compliance, and second is to reduce the surgical cost. Third, to improve the post-surgical recovery and reduce stress. And option D is to increase the number of experiment per day.
Is it option C?
Yes, the right option is option C. Can you little bit explain about it?
Yes, ma'am. Since like we are not invasing something or
Hmm.
implanting something inside,
Hmm.
so it will like create. I mean, the recovery time for any kind of minimally invasive surgeries will be much lesser than an invasive technique. And since like it is minimally invasive, the stress experienced by the rodents will also be less.
Yes. So the right option is option C. Okay. So next, coming to the question number 18. What is the one of the key factors to consider when the designing implantable devices for rodent? First, the weight and size of the device related to the rodent. Second, the aesthetic design of the device. Third is the manufacturing cost of the device. And fourth option is the availability of the device material. So go through the question carefully and try to give the right option. The right option is option A, the weight and size of the device related to the rodent. Okay. So next move toward the question number 19. Which of the following is the potential biomedical application of the implantable devices? First, tracking the exercise performance. Second, continuous glucose monitoring. Third is enhancing the MRI imaging. And fourth is studying the interaction implant.
Ma'am, is it option B?
Yes, continuous glucose monitoring. Yes, the right option is option B. Good. So this is the question number 20. Why is the use of microsensor growing in the field of biomedical research? First option, they are inexpensive and widely available. Second, they allow the precise and real-time measurements in small area. Third, they eliminate the need for ethical approval. Fourth, they replace the traditional surgical procedures. So which one is the right answer?
Ma'am, option B.
Yes. Can you a little bit explain?
Yes, ma'am. Since the implants contains microsensors, which are which we or the rodents are carrying, so
Hmm.
we can have the real-time measurement of any signals that we want to detect. So that's why, and definitely it's much more precise than
Yes,
traditional.
the small areas. Yes. So the right option is option B. They allow the precise and real-time measurement in a small area. That's why we're going for the micro, right? There's micro where the everything is in the micrometer range. So next question is the question number 21. What is the cryonotomy as described in the rodent neurosurgery? So first is the removal of brain tissue for analysis. Second, the surgical removal of part of the skull to access the brain. Third is the implantation of electrodes for neural recording. Fourth, non-invasive imaging of the brain. So go through the question carefully and find out the right answer.
Ma'am, it's option B.
Option B, surgical removal of the part of the skull to access the brain. I think they have in the lecture 3 they have shown how they are deep into the brain. There is a certain layer is there, and how the surgical how they are going to the deeper layer. The surgical removal of the part of the skull to access the brain. So this is called the cryonotomy. Okay. So next move toward the question number 22. Which part of the rodent brain is commonly targeted in neurosurgical studies for memory and learning? First, the hippo.
Hippocampus, ma'am.
Yes, what is the right answer?
Ma'am, it's hippocampus.
Okay. Anyone else? Nikita, Shushi, and Gururaj. Anirudh. What is the right answer? Yes, obviously this right answer is the hippocampus. Okay. Next, move toward the question number 23. Why is rodent neuroanatomy considered crucial for the biomedical research? Rodents have the similar brain structure to human. Rodents are less expensive and easier to handle than primates. The third is the rodent models allow the detailed studies of neurological disease. And option D, all of the above. It is the right answer.
Answer D, all of the above.
Yes, answer D. Can you little bit explain? Yes, your answer is right. Little bit explain.
As we studied in these lectures, we
Hmm.
got to know that rodents and humans have similar brain structures.
Hmm.
So option A is correct.
Okay.
And obviously, rodents are lesser expensive and easier to find
Hmm.
in the environment. And Rodent model allows detailed studies of neurological diseases because of the similarities in the brain structures
Yes.
and even disorders.
Okay. So the right option is option D. So next, good. Next, the question number 24. What is the main function of the cerebellum in rodent and the humans? First, the control of emotion. Second, the coordination of voluntary movements. Third is the regulations of hormones and hormone release. And fourth option is the processing sensory input. So what is the right answer? Cerebellum.
Ma'am, is it coordination of the voluntary movement?
Yes, yes. The right option is the option B, the coordination of the voluntary movements. Okay. So next we move toward the question number 25. What is the purpose of implanting the electrode in rodent brain? So first option is the to stimulate the neural activity. Second, to record the electrical signal from a specific brain region. Third, to treat the neurological disorder. And the option D is both A and B.
Ma'am, it's option D.
Yes, option D, both A and B, to stimulate the neural activity and also to record the electrical signal from a specific brain region. Okay, so next move toward the question number 26. Which type of microsensor is commonly used for monitoring the neurotransmitter level in the brain? First option is the optical sensor, second, the electrochemical sensor, third is the magnetic sensor, and fourth is the thermal sensor. So, what is the right answer here? They have discussed in the lecture which type like different of sensor they have put it in the forehead. So they have used a term that the AGCL electrode. Have you heard about this word? Hello.
No, ma'am.
In the lecture they have also discussed about it, and it is a part of the electrochemical sensor. Okay. So the right option is option B, the electrochemical sensor. So next move toward the question number 27. In comparative neuroanatomy, what is one major structural difference between the rodent and human brain? So first is the absence of a cerebellum in rodent. Second, the larger reactive size of the olfactory bulb in rodent. And third is the presence of gyri and sulci on only in rodent brains. And the fourth, a more developed limbic system in rodent. So go through the question carefully and find out the right answer. So what is the main difference here they're asking about?
Ma'am, the correct answer is option B.
Okay. The larger reactive relative size of the olfactory bulb in rodent. Yes. The right answer is option B. So next move toward the question number 28. Which brain region is commonly associated with the motor control in both rodent and humans? First is the basal ganglia. Second is the amygdala. Third is the hippocampus, and fourth is the occipital lobe. So go through the question carefully and find out the right answer. The right option is option A. Okay, the basal ganglia. So next question number 29 is what is the primary advantage of using the chronic implants in rodent neurosurgery? First, avoiding the need for repeated surgeries. Second, the improving the aesthetic appearance of the rodents. Third is the allowing the multiple animals to be studied simultaneously. And third is reducing the cost of surgical procedures. So please try to find out the right answer.
Ma'am, is it option A?
Yes, option A is the right option. Avoiding the need for repeated surgeries. Good. So, next question number 30. What is the stereotoxic surgery in rodents? First, a non-invasive imaging technique. Or second, a method to accurately target a specific brain region using a 3D coordinate system. Third, a technique for removing brain tissue. Fourth, a method for placing implant in a special spinal cord, in the spinal cord. So, what is the right answer? So, what is the right answer here?
Answer B.
Yes, the right answer is option B. A method to accurately target a specific brain region using a 3D coordinate system. Good. So next question, question number 31. What is the function of cerebrospinal fluid in the brain? First, to transport the oxygen to neurons. Second, to cushions the brain and spinal cord from injury. Third, to carry the electrical signal between the neurons. And the fourth option, to regulate the blood pressure. So, what is the right answer? So, what is the right answer? What is the function of CSF in the brain? To transport the oxygen to neuron, to cushion the brain and spinal cord from injury. Third, to carry the electrical signals between the neurons. Fourth, to regulate the blood pressure. The right option is option B, to cushion the brain and spinal cord from injury. Okay. So next move toward the question number 32. Which of the following is a key consideration during the rodent brain surgery? First, the precision in targeting the brain region. Second, to maintaining the sterile conditions to prevent the infection. Third, minimizing the tissue damage. Fourth, all of the above. So find out the right answer.
Ma'am, it's option D.
Can you explain a little bit?
Yes, ma'am. While we are doing this brain surgery in rodents, we must be very much precise towards the target area, like which part of the brain we are actually targeting.
Mm,
Secondly, sterile condition is always a first-hand goal for any kind of surgeries, just to prevent infection. And while we are doing the surgery or we are clipping the brain regions, we should be very much careful about the tissue damage. We should minimize it as much as possible.
yes. So the right option is option D, the all of the above. Okay. So next move toward the question number 33. Which brain region is involved in emotional processing and is studied in rodent neurosurgery? First is the amygdala, third is the thalamus, and second is the thalamus, third is the brain stem, and fourth is the cerebellum. So what is the right answer here? So the first right option is option A, the amygdala. Okay. So next move toward the question number 34. Which imaging technique is often used to visualize brain structure in rodent before surgery? First is the X-ray, second is the MRI, third is the ultrasound, and fourth is the PET. So what is the right answer here? For before the brain surgery, what type of techniques we have used? Which imaging technique is often used to visualize the brain structure in rodent before surgery? So the right option is the MRI. Okay. So we have gone through the MRI, magnetic resonance imaging. So is this the next question, the question number 35. Why it is important to compare the neuroanatomy across the species in neurosurgery research? First, to understand the species specific behavior. Second, to adapt the surgical techniques for human applications. Third, to explore the evolutionary development of the brain. And fourth option is all of the above. So please go through the question carefully and find out the right answer. Dipstuti, what is the right answer?
Mm, Ma'am, is it all of the above?
Yes, the right option is all of the above. Can you please little bit explain?
Yes, ma'am. Across the species, when we are talking, so like to understand specifically each species behavior, we do an across species study, and
Yes.
like to adapt surgical techniques for human applications. Like before we apply something to humans, we have to go for trials and also across species, whether it may be rodent or primates, we prefer doing it at first on them, and by studying like neuroanatomy across species, we get to know about the evolutionary development that has happened in the score of time.
Okay, good. The right option is option D, all of the above. So this is the next question, question number 36. So what is the primary goal of micro neurosurgical instrument and technique as discussed in the course? So first option, to perform the neurosurgery on humans. Second, to develop tools and methods for precise surgical um, interventions in small animal models. Third, to create the robotic system for surgery. Option D is to study the history of neurosurgery. So go through the question carefully and find out the right answer. One more question is left. So what is the right answer? Devstuti, what is the right answer?
Is it option B?
Yes, option B is the right answer. To develop tools and methods for precise surgical intervention in small animal model. Right option is option B. So this is the last question, the question number 37. Why it is important to understand the comparative neuroanatomy in the context of rodent microsurgeries. First option, to develop a new surgical instrument. Second, to apply the finding from the rodent model to human neurological condition. Third, to create a more effective anesthesia protocol. And fourth option is to design the better laboratory environments for the rodent. So please find out the right answer.
Answer B.
Yes, answer B is the right option. To apply findings from rodent model to human neurological condition. So we are ending our session here. Next we meet next Tuesday 7 to 9 p.m. Okay. So you can leave.
Okay, thank you, ma'am.
Uh,
Thank you, ma'am.
okay, okay. Welcome.